With each puff, a cigarette smoker inhales over 60 known or suspected cancer-causing agents, carcinogens, including polyaromatic hydrocarbons, PAHs, nitrosamines, and heavy metals. Smoke moves with inhaled air down the respiratory tract, from the trachea to the bronchi, and then, branching into ever smaller bronchioles. The bronchioles end in alveoli sacs, where nicotine, carbon monoxide, and other gases in cigarette smoke are exchanged with the blood. Smoke particles and gases are trapped in mucus that lines the cells of the respiratory tract. Hair-like projections, called as cilia, beat to sweep particles out of the lungs. Smoke slows down and paralyzes cilia, impairing the lungs' ability to detoxify. Years of smoking eventually destroy cilia completely, and the lungs lose their sweeping effect. Then, cigarette particles become trapped in the mucus and cannot be expelled. A thick, brownish tar builds up in the lungs, giving them a dark color. Carcinogens can then enter the lung cells and cause DNA damage. The damaged cells may eventually progress to lung cancer. K-ras and p53 are the two genes most frequently mutated in smoking-related lung cancers. One tar component, benzo, pyrene, is specifically linked to known mutations in these genes. Within a lung cell, benzo, pyrene is converted to an epoxide. The epoxide reacts readily with guanine positions of the DNA helix. If not corrected by the cell's DNA repair mechanism, this guanine adduct is misread as a thymine by the DNA polymerase that copies chromosomes during replication. Ultimately, the original GC base pair may be replaced by a TA base pair, a mutation called a transversion. Cultured cells treated with benzo, pyrene show the same spectrum of GT transversions as found in the K-ras and p53 genes of smokers. These mutational hotspots map well to the guanine binding sites of benzo, pyrene epoxide. Benzo, pyrene can produce the major known activating mutation in the 12th codon of the K-ras gene. Benzo, pyrene can also mutate 3 key positions in the p53 gene. The protein produced by the K-ras gene is a tumor activator. K-ras is analogous to a car accelerator because its overactivity contributes to tumor development. The K-ras protein resides on the inner side of the cell membrane, where it conducts growth signals from cell surface receptors to the nucleus. This process is called signal transduction. Signal transduction begins with the arrival of a growth factor at the cell surface, where it recognizes a specific receptor anchored in the cell membrane. The binding of the growth factor to its receptor conducts a growth signal into the cell interior. The K-ras protein accepts the growth signal and, in turn, relays it to other molecules in the cytoplasm. RAF and other signal transducers are protein kinases, which activate other molecules by adding phosphate groups. This signaling cascade culminates in the nucleus with the activation of Phos and Jun, 2 transcription factors that join together to initiate transcription of genes involved in cell replication. Mutations in the K-ras gene result in a K-ras protein that is essentially stuck in an on position, perpetuating a signaling cascade in the absence of any real signal from a growth factor. Mutations in the p53 gene are found in 70% of lung tumors, the highest rate for any cancer. The p53 protein is a tumor suppressor, analogous to car brakes, because its activity helps counter tumor development. p53 occupies a checkpoint in the cycle of cell division, where it senses DNA damage or mutations. The cell cycle is composed of 4 stages. During the first gap phase, G1, the cell grows and replenishes its resources. During S phase, the cell synthesizes DNA in preparation for cell division. During the second gap phase, G2, the cell synthesizes proteins and other cellular components needed for cell division. During mitosis phase M, the cell divides into 2 daughter cells. p53 acts as a checkpoint into the critical synthesis, S, and mitosis M phases. After receiving information from DNA repair systems, p53 can signal the cell to stop dividing, allowing time for a mutation to be repaired before it is passed on to daughter cells. For example, p53 arrests the cell cycle, allowing time to repair GT mutations induced by benzo, pyrene. If the DNA damage is too great to repair, P53 can signal the cell to commit suicide by the process called apoptosis, or programmed cell death. Mutations in P53 cause a loss of checkpoint control, allowing mutations and DNA damage to accumulate in a cell lineage. Nicotine has long been known to be the habit-forming drug in cigarette smoke. However, recent research shows that nicotine also works with other components of smoke to promote cancer formation. So there are really three principal components of tobacco that have been identified that have adverse biological effects. Those include carcinogens, such as polyaromatic hydrocarbons and nitrosamines, as well as nicotine. The classic mechanism of lung carcinogenesis is based on the fact that carcinogens, when they are activated, end up causing DNA adducts and DNA damage. Many of the nitrosamines that are carcinogens are actually nicotine metabolites, but nonetheless, they are present in cigarette smoke. So that when a person smokes, there is exposure of epithelial cells to nicotine and nitrosamines. Nicotine and nitrosamines bind to nicotinic acetylcholine receptors, which have recently been described on epithelial cells, which then transduce a signal to intracellular kinases that have profound cellular effects. So the serine-threonine kinase act has become a very hot molecular target in cancer biology. It is a kinase that is activated in response to many types of stimuli. Once active, ACT will phosphorylate many downstream substrates. Over 50 have been described to date, and some of them are very key players in control of cell cycle, such as P27 and P21, apoptosis, such as BAD and MDM2, and protein translation such as mTOR and TSC2. When ACT is activated by tobacco components in normal cells, it leads to the increased proliferation and survival of these cells. In addition, active ACT has been detected in the precursor to cancerous lesions, bronchial dysplasia from smokers. So the cumulative evidence suggests that ACT is an early and important target in lung cancer formation. ACT likely plays a role in lung tumorigenesis through the following mechanism. A smoker is exposed to nicotine, which, although we know it has biologic effects outside of addiction, is the addictive component. This leads to exposure to the carcinogens that cause DNA damage. If DNA damage is not repaired, the cell has a crucial decision to make as to whether or not to undergo apoptosis. The role that ACT plays is probably in that crucial step. Because tobacco components activate ACT, which inhibits apoptosis, if ACT is active, the apoptotic threshold is altered. And if apoptosis does not occur, it can lead to the accumulation of genetic changes, such as K-ras mutations, P53 mutations, etc., that are necessary for lung cancer formation.